Hi, my name is Deb Kramer. I'm the founder and executive director of Keep Coyote Creek Beautiful, and I'll be your moderator tonight. I love exploring and learning about nature. And over the past 11 years, I've had the great fortune to build a community around Coyote Creek through our events. Through my work along Coyote Creek, I've met many incredible people, and in this presentation, you'll meet a few of them. So for today's training, I wanted to know, are you curious about the wildlife that comes alive after dark? Well, in this webinar, we will explore nighttime adventure, exploring the fascinating creatures of the night. During the webinar, Dave Johnston will present the incredible ecology of California bats, and Jack Owicki will introduce critters that shine in the dark with a little help from UV. Together, they'll review a hidden nighttime world that many of us rarely get to see. Finally, we'll close with time for questions and answers. So let me tell you a little bit about Coyote Creek. Coyote Creek is a 64-mile river starting in Henrico Park, and it winds through Coyote Valley, through downtown San Jose, into Milpitas, and finally into San Francisco Bay. It has a huge diversity of wildlife and ecosystems. Next slide. Here you can see the map of Coyote Creek, where the Diablo Range is on the east side of the creek, and the water flows towards the middle and pretty much is down along Highway 101. Next slide. Keep Coyote Creek Beautiful was founded by me in 2015, and we basically do three things. We care for the creek and advocate for its health. We educate people about the value of having a healthy watershed, and we also offer recreational activities to connect you with the watershed. Next slide. We like to say that it's all connected. We engage people to provide a voice for the creek and pick up trash. We encourage folks to get out and participate in fun activities, get some fresh air, a little recreation, and meet some new people. And then we provide education where people can learn about the watershed, whether they're children or adults, and they can see what's so special in this urban landscape. As you can see from the next slide with the images of people enjoying nature, we really focus on building our community with nature as a center point. Next slide. And we've held a naturalist training series. We've had 13 events, and you can learn more about them on our YouTube channel at Keep Coyote Creek Beautiful. We have a few upcoming events that Keep Coyote Creek Beautiful is hosting, some with BioBlitz Club. We have a creek cleanup in the beginning of April. We also are participating in the City Nature Challenge with a BioBlitz at Alum Rock Park, followed by a BioBlitz at Hellier Park in May. And in between, as part of the City Nature Challenge, we're hosting an ID party. All right, if you can unshare now, what I'll do is introduce Merav Vonshak. So Merav founded the BioBlitz Club, which is a community science organization to encourage people to get outside and learn about nature. Through this group's actions over the past 9 years, we've mostly been doing this together for those 9 years. She has hosted nearly 160 events with thousands of people who participated and contributed to the growing body of knowledge about nature through iNaturalist. All right, take it away, Merav.
Thank you, Deb. Okay, let's do it. So hi everyone, my name is Merav, and I just want to quickly share just a few of my upcoming events. You could join a bunch of BioBlitz events, many with Keep Coyote Creek Beautiful and some with other groups. You can find everything on bioblitz.club. Okay, but now we're going on a different adventure. We're going on a night hike. And there's something really special about going out at night. And hopefully by the end of this presentation, you'd want to just go out with a flashlight and just look for stuff. But even without a flashlight, just being outside at night is an amazing experience. So let's just take a listen for a second. So I hope you were able to hear all these beautiful frogs calling in the background. I took this recording at some night hike a few years ago. Yeah, so when you go outside, you can't see much unless you have your flashlight on. But it's a really interesting experience to just listen to all these different calls that you could hear at night. Um, and I think it's a special experience also for kids, even if you're just going camping, you could just explore your campsite area, or even if you're home, just go outside and take a walk on the sidewalk in your street and see what you can find. Because once you start looking, you'd be surprised what you could see at night. It's just amazing. All these beautiful creatures that go out at night. And if we're not there, we wouldn't know that they were out. And of course, it's always good to have the right tools. So the Ecometer bat detector is an amazing gadget that you could add to some smartphones. And then having a really good flashlight with rechargeable batteries and a UV light that Jack will explain more about later. These are really good tools. And also a moth light. So by attracting moth and many other insects, we could see what's out there in a specific area. You could do it in your backyard, on your balcony, or when you travel, and you'd be amazed at the stuff you can find. And I also quickly wanted to mention my group, the Newt Patrol, where we go out and document newts, but we also started an assisted migration program where we go out at night and we help the newts cross the road. And it's marvelous. And we also have frogs calling in the background. So it's just beautiful. And I'd invite you all to join us.
Jack Owicki is a retired biophysicist who normally spends his time as a docent at Mid-Penn Regional Open Space District and Stanford's Jasper Ridge Biological Preserve, or wandering trails and taking photos of little things. He's especially fond of arachnids. He dedicates most of his time helping to run ProBonoPhoto.org, a grassroots photojournalism group.
Thank Deb and Merav for inviting me just to talk about fluorescence out in nature at night. I've got two sources of my interest in this. In my scientific career, I used fluorescence as an analytical tool, used it a lot. So I'm kind of familiar with the nuts and bolts of it. And then in my docenting, I've been docenting at Mid-Penn Open Space Preserve for 15 or 20 years. And early in that, I started to specialize in night hikes and often went with Debbie Briscoe, whom I think some of you know. And we early on got interested in fluorescence, carrying what tiny UV flashlights were available then. Things have changed a lot, but it kept up on this. Let's see here. Just a second. These things never seem to go easily. There, okay. So first I'm going to talk about what fluorescence is. And fluorescence doesn't necessarily involve ultraviolet light, but when ultraviolet light is used to get fluorescence, it has lots of advantages if you're out in the wilds looking for things. And I'm going to talk about that and what equipment you ought to get. First thing to get straight is bioluminescence and fluorescence often get confused. In both cases, you have something glowing in the dark, but the source of energy that drives the glow is different in the two cases. For bioluminescence, the source of the energy is a chemical reaction. It's like, the glow sticks you break and start the chemical reaction. But for fluorescence, the source of the energy is light that you shine on it. The slide here is one of our few bioluminescent critters around here, the California pink glowworm. And I'm going to speed things up a little bit because we're late, so I won't talk anymore about this one. Just to get us on the same page, I'd like to briefly review the relationship between ultraviolet and visible light. So you've got the spectrum here from in wavelength from about 300 nanometers up to 700 nanometers. And here is the Roy G. Biv going in that direction. And then there's a UV somewhere in the neighborhood of 400 nanometers. You switch over from UV to visible. One thing to note here is that this side, the short wavelength stuff, is very high energy. You get sunburns from UV, not from visible light so much. Over here at long wavelength, it's low energy, and that will turn out to be important. So here's how the fluorescence works. Let's say that you have a molecule. The circle's a molecule, and let's say it can absorb blue light. Different molecules absorb different colors of light. Right? This one's going to absorb blue. So you shine blue light on it. What happens? Well, a lot of the light is still going to be reflected and scattered, and that's it for the light. But some of the light will be absorbed. There's energy in the light. So now, at this point, the molecule is really jazzed. It's highly energized, and it will, before too long, return to its original state by getting rid of the energy. And the important thing here is how it gets rid of the energy. There are multiple pathways, multiple processes that occur in parallel, and they kind of compete for the energy. One of them, the most common one, is just dumping that energy as heat. And you go out into the sun and you get hot. That's what's going on there. One of the other possibilities is that the energy is converted into light and is emitted. That's fluorescence. The significant thing here is, though, that in that process, there's a little bit of heat generated. So you can see kind of an equation. The energy of the fluorescent photon plus the energy of the heat that's left has to equal the energy of the photon that went in, right? So this has to have, the fluorescence has to have less energy per photon than the light that went in. And what that means is that, if you shine light, say blue here, and you get fluorescence, the wavelength of the fluorescence has to be somewhere to the right, to the lower energy. If you shine UV, you hope that you get fluorescence over here somewhere. Now, let's see. Most organisms are not fluorescent. And therefore, those that are fluorescent really stand out. Why would things not be very fluorescent? Well, for one thing, looking over here, if you shine light on something, it can't emit fluorescence if it can't absorb the light. So you pick a wavelength of light to shine on it. Only things that absorb that will ever have a chance of doing fluorescence. Everything else will be dark. The other thing that controls this is, as I was talking about before, there's a competition between just dumping the excited energy to heat and doing something nice like fluorescence with it. And for the vast majority of molecules, and molecules are just embedded in the skin of or the cuticle of an arthropod or in the tissue of a plant or something like that. For the vast majority, this process of dumping the heat is so fast that poor fluorescence never has a chance. There's a small number of cases where the fluorescence is fast enough that you actually get an appreciable amount of fluorescence out. But fluorescence is usually very dim. And so if you have any ambient light around, like daylight, it's just swamped by the other things that are going on in the daylight, and you can't see it very well. So nighttime is an ideal time to be looking for fluorescence. And also, if you are, even in the night, if you're shining blue light, say, on it and getting green fluorescence, there's got to be a whole lot of blue light bouncing around. It still may be hard to see the fluorescence, even if there's no sun shining on it. So what if you couldn't see the light that you were using to get the fluorescence, but it's still there? That's ultraviolet. So ultraviolet light you can't see the light you're shining on it. You do it at night, there's nothing else, and fluorescence just pops out if it's there. What kind of you need a flashlight? What kind of flashlight should you get? They're going to be based on UV LEDs, ultraviolet LEDs, and they're widely available online. They have rechargeable batteries. Almost all of them will take rechargeable batteries. And how much you have to pay depends mostly on the brightness that you want. Typically, the brighter they are, the more you have to pay. But I would say that for purposes of being out in the wilds or field natural history, if you spend about $15 on the right one, you can get something that'll be fine up close. If you want to see things far away, a lot of brightness, you need to pay more money. And they're available in different wavelengths, essentially different colors of ultraviolet, from 365 nanometers up to 395 nanometers. And you always want to buy the 365 model. And that is because, so here's the spectrum again. So the border between what you can see and what you can't see is right around somewhere between 380 and 400 nanometers. It's not like you can see it, and then suddenly nothing. It's a gradual fall off over this period. So if you use a UV light that's essentially right over here. And the UV lights are not lasers with only one wavelength. They're a band, maybe 20 or so nanometers wide. So if you start to shoot UV light in that's close to this, it's going to spill over into the visible range, and you're going to see violet, a violet background, which is just the light from your flashlight. But if you choose a UV light that is far enough into the UV, then you don't get this spillover. And here's a concrete example of that. 2 scorpions, okay? The good one here is illuminated with the 365 nanometer light. The other one here is illuminated with a longer wavelength light. And you can see, yeah, you get fluorescence, but look at all of the ugly background that you get too. Not as good contrast here. Okay, so now I want to talk about fluorescent living things that you might see in Northern California. And I'm going to have some arthropods, like I was kind of intending to, but I'm going to go much wider than that. Just give you a quick menagerie of things you might see. Okay, first of all, scorpions are the poster children for this. People have been looking at scorpions with ultraviolet, mostly out in the deserts at night, for ages and ages, but they had very expensive mercury vapor lights and things. Now you've got these cheap, relatively cheap UV lights. And you can see if the difference between being able to see the scorpion.
We can't see the slides. We can only see
okay.
how fluorescent works.
Okay. Sorry about that. There. How's that?
And by the way, Jack, I'm just going to give you another 5 minutes.
Okay.
I know we got cut off, but I want to make sure that Dave gets some time too.
I shouldn't need much more than that. I can just run right through these. You can see scorpions are well camouflaged around here, but not if you show them, if you have UV. Some spiders and harvestmen are really nicely fluorescent. We have 2 species of millipede that are fluorescent here. During the cool, wet months, you see the millipedes. During the warmer, dry months, you see the scorpions. And here it is in regular light, and here it is in UV light, even with some visible light on it. Here is a couple of mating. And here, if you can see the crawling, watching them crawl is really a cool thing. Second type of millipede we have around here, yellow spotted. Here it is in regular light, and here is a picture by Merav in UV and regular light. Leaves, chlorophyll fluoresces red. If the leaf is really functioning well for photosynthesis, the photosynthesis gets the energy before it can fluoresce, and you don't see fluorescence. If it's not an actively fluorescing leaf, you get red. Here's a polypody fern with sorry that's not actively fluorescing. Flowers, some of them are really cool. Dandelions, very nice. Fetid adder's tongue, not so cool, but it's something. Fungi, a lot of them fluoresce. Sulfur tuft over on the left, a very common mushroom here, green under the gills. Here's a fungus that is digesting a leaf, and right where it's actively digesting, there's a lot of fungus, and it's got yellow fluorescence. Lichens are great. You go down the trails here, you can see lichens that fluoresce blue, green, yellow, orange, and red, all sorts of colors. Here's one that's naturally or yellowish lichen, but with UV illumination, it turns beautiful red. There's a lot of fluorescence in the ocean. If you want to go tide pooling at night and don't worry about waves that you can't see, you'd probably see a lot of things. Here's a jellyfish and an anemone that both fluoresce. Lots of other things do too. The brightest fluorescence you'll see of all is going to be human trash. And it's usually very bright blue fluorescence. It'll be a piece of white paper or maybe some fiber from fabric. And this is some fabric fiber, so it's the brightest thing around. The last thing I want to do is to introduce mammals. It's been found in the past, 20 years or so, that the hair and skin of some mammals fluoresces. And some bats do that. Here's a museum specimen of bat that was illuminated with violet light, and they took a picture, and you see there's a lot of violet light on that. If you screen out all the blue and the violet light from that picture and do it again here, you see the green fluorescence. Also, many of you know about white nose syndrome in bats caused by a fungus, and it causes skin lesions. And those skin lesions fluoresce yellow-orange with UV illumination. So here's a wing stretched out of a bat that's afflicted with it, and all of these yellow spots are the fungus, the white nose fungus. And this could be a diagnostic tool in the field. And that's it. Thank you.
I would like to, at this time, introduce another colleague, and that is going to be Dave Johnston. So Dave has studied the conservation and ecology of bats for over 30 years, and he currently has ongoing bat projects in California, Belize, and Costa Rica. He serves as executive director for the Youth Science Institute in Santa Clara County and lectured at Santa Clara University. Enjoy his presentation about the wonderful world of bats.
Thank you, Deb. I will go straight into the program here because we have limited time. So without further ado, we're going to go ahead. So a lot of, for many, many years, particularly in this country and in Western culture, bats have had a bad rap. And a lot of people still think that, most bats have rabies. They do not. They have the same incidence of rabies as any mammal. It's about 0.5% in the natural population. They get tangled up in your hair. Well, people are afraid of bats, but it's because of the culture that we've brought up in. And in some parts of the world, like China, the emperors always had bats embroidered on the outsides of their robes. And if you look at a lot of the Chinese china, stoneware or porcelain, and I have a couple of sets that have little bats on the edge of them. They're stylized, but if you look carefully, they have bats which symbolized longevity and good health and prosperity. So I want to go through, and we're going to talk a lot about really bats that you might find along the Coyote Creek. Now, when I grew up, I thought of bats as these black things that, just sort of, flew around in the sky. I did not know much about them, but, I just knew that, we didn't know a lot about them, but they're all dark or black. And when I started studying bats, I thought, wow, the diversity is amazing. So I took this picture in Costa Rica some years ago, and it's a tent-making bat. It hangs from a leaf of a heliconia like a banana leaf. And it was sleeping, and then I looked up and I took a picture of it. It woke up and it just looked straight at me, and I looked straight back, and I got a couple more pictures, and then I left and it went back to sleep. But there are now over 1,500 species of bats that are recognized by science. And we are finding new species every year. So I want to take you through just some of the diversity, some of the things that bats eat, and then we're going to concentrate on some of our local bats here. There are no bats that are blind. Even this bat, which has very tiny eyes, this is a horseshoe bat, and yes, it hunts primarily through echolocation. It uses its large ears. It probably doesn't have very good vision, but in fact, it does see. And some bats have very, very large eyes and big noses and small ears. This is one of the flying foxes or pteropods. It's an epauletted bat from Africa, and it finds its fruit primarily by what? Smell, of course, like a dog, and it's often hunting for its fruit in the early evening. So it's crepuscular, meaning that it uses the twilight. The largest bat in the world is the gigantic flying fox from northern India. It has a wingspan of about 2 meters, or really probably closer to 6 feet. And this is, again, another tropid. It does not echolocate. It has small ears, does not have a tail. And the tropids, or the flying foxes, which is a separate suborder, has a small nails on its fingers. So most bats just have a nail on their thumb, but the tropids also have this little nail on their index finger. So bats are basically flying with their hands. And chiroptera means chiro as in chiropractor, someone who practices medicine with their hands. And ptera, we all think of the pterosaurus or pteranodon flying lizard. So bats' scientific name, chiroptera, is really flying hand. Okay, and some bats are just incredibly beautiful. I mean, in terms of their complexity. So this is a wrinkled-faced bat. It's a very interesting bat because it will fold its wing up over its head when it's disturbed or when it doesn't want to be found. And the wing is so thin and transparent that it can probably see motion, but there's no eye shine, which is really important if you want to evade predation. And for years, scientists have contemplated, debated, discussed why they don't have any fur on their face. This is the eye, by the way. Their nose is down here, the mouth below that, and this is the bottom of their ears. That's the little pinnae right there. I mean, the tragus. The pinnae is the fleshy part of the ear. So we think that it's probably because they put their heads in very fresh fruit, and it's gooey and gunky, just as a vulture might do that in a carcass. But the vulture is, in gunky flesh, whereas this bat is probably in gunky fruit. The bats that we have here in the Bay Area are all insectivorous, as this spear-nosed bat is. And this is a yellow-shouldered bat, Stenira lilium, but The neat thing about this picture is you can see on the top of the head there, the pistil, the female part of the flower. And it already has pollen on the head. So when it's visiting this ocotillo flower, then it's actually pollinating flower by flower and presumably plant to plant. And here's another yellow-shouldered bat. It's pollinating this banana. I'm going to go right back to this for a second. Excuse me. The very cool thing is that the banana plant has bracts, and these bracts open up to perfect parabolic mirror-like shapes that make a hot spot when the bat is echolocating. So the bat echolocates into the banana, and when that bract is perfectly shaped and it makes a big white spot in terms of a echolocation response, the nectar is flowing and the pollen is ready to be picked up by the bat. And some bats, like this Stracops, is eating a frog. The frog, that's a male frog. When it calls, it doesn't know it's also alerting the fact that it's, in a position where one of these frog-eating bats can take it. I've studied fishing bats in both Mexico and Belize and Costa Rica. This is the greater bulldog bat, and it has claws or talons that are about a centimeter long, razor sharp, and it simply glides those through the water after it detects a small amount of disturbance in the water. And finally, this is the greater false vampire or Vampirum spectrum, and it's simply yawning. But the head is about the size of a fox. It's the largest New World bat, and it has a wingspan of about 3 feet, and it is a serious carnivore. I mean, it'll take out chickens, rats, other bats, and so on. And you can think of them as sort of the mountain lions of the bat community. Okay, a little bit about sonar. So we could spend a long time just on sonar, and it's one of the nice things you can do at night in the field with any number of instruments, some more expensive than others. I think for a couple $100 now you can get a little attachment to your phone from Wildlife Acoustics. And it is not very accurate at determining what bat you're hearing, but the microphone is good, and you can use other programs to do a little better job of identifying the bat. But here the bat is putting out a high frequency sound. It comes out, it hits, this is a spingid or hawk moth, and then it comes back, a small amount of that bounces back, hence the name echolocation. It's locating by the echo. Now, this is so intense that if you had a smoke detector alarm 1 inch from your ear, that would be about the same volume of sound, the same decibels, which would be deafening. And over time, you would probably go deaf. So what the bat does, which is like this amazing evolutionary development, the bones in the inner ear disarticulate. They actually come apart so that there's no noise that or sound that is received by the bat the instant the sound goes out. So the bones disarticulate and the sound goes out, then the bones come back together and it hears the sound. And, bats are doing this like in milliseconds a time. And so, it's a really rapid, very rapid movement. Okay, whoops. Okay, so I want to talk about our own bats here along Coyote Creek. And the two most common bats that we have are the Mexican free-tailed bats and the Eumamyotis. And many of the other species are in decline. And I would say all the other species we have in California are in decline. But these two are on the increase. And I will show you why. One of the things that I believe has increased both species is that when we built buildings, this is the Mission Dolores, 1776 in San Francisco, and they use tile roofs. Tile roofs make great crevices. They warm up during the day. They stay warm through the night. Perfect place to raise young. Another thing that we've done is at metropolitan areas, we've built huge dams and reservoirs. The dams have acted as sites where a lot of bats can roost during the day, and the reservoirs have greatly increased the foraging habitat of both those species. In fact, the Eumamyotis has specialized on flat water habitat, and we have so much more of that today than we used to pre European colonization. So what does urbanization do? Besides increasing those two species because we have more habitat, most of the other species have declined, particularly as a result of urbanization, noise pollution, light pollution, and probably most significantly, habitat conversion and destruction. So I listed here the bats that you now find in Marin County, which still has some wild areas, 13 species. If you go into the virtually totally urbanized San Francisco County, only 4 species of bats, and of these, primarily only the first two, western red bat and little brown bat, come through the area, but only as migrants. Okay, a little bit more about Eumamyotis. I want to tell you where you can find them. They forage not only on flat water all along Coyote Creek, but also in the South Bay. I had a graduate student put out bat detectors for her master's over much of the saline and brackish water, and they regularly forage over insects that tolerate brackish water. And they also go over pure marine areas where you have flying insects. But they're primarily a emergent aquatic insect forager. So when you go out and you see a bat fairly small, flapping pretty hard, that's almost always a Eumamyotis if it's flying along the water. The Brazilian free-tailed bat, or as we sometimes call it, the Mexican free-tailed bat here, because that's the subspecies for Western North America. They fly much, much faster. They have a low, if you're using your bat detector, they have a very low echolocation of around 20 to 30 kilohertz, whereas Eumamyotis is at 50. But these are amazingly eclectic and flexible in terms of what they eat and where they go. So they follow moth migrations in California as they move north, and they are found in every one of our habitats and up to about 10,000 feet. So I want to show you a couple of videos. This is of a cave in Texas. Whoops. This worked before. There we go. Just amazing. So at this particular cave, there are over a couple million bats emerging. The closest thing that we have here is Yolo bypass. I think it probably has about 250,000. And I have been counting the Franklin Boulevard Bridge bats for 15 years now, and they're about 160,000. Okay, I'm going to do this one. When I show this to kids, I ask them to count the bats. And then they say that you can't do that. And I say, okay, well, then we'll just slow it down for you. And it's going to slow down here. There we go. Now count the bats. We have video equipment now that can make a polygon around each one of these images. And as it separates from another image, it will count the bat. But we don't do it long ways. We do it on the side. And it's probably as accurate as trying to count them just by our own eyes. Okay, I have caught the hoary bat both along Lower Coyote Creek and then in Penitentiary Creek. And this bat is found throughout North America. It is an incredible flyer. For about 10 years, I worked on the Hawaiian hoary bat, which is sister taxa to this, or taxon to the mainland hoary bat. But they are, I think, in serious trouble because of the wind energy turbines. And there are at least 7 hypotheses why they get hit. But for a number of reasons, they are probably attracted to wind turbines. And we're still trying to figure out exactly why and how we can reduce that. There are things that we've done that will reduce it a little bit, like broadband high frequency sounds, UV lights, but in the end, we're still killing a lot of hoary bats, which is just awful. So they are solitary bats. They roost in foliage. They don't roost in crevices. And they're solitary. They'll have 2 young every year. But they can fly probably 60 miles an hour. At least that's what we found in Hawaii. Okay,
Dave,
yes.
you've got just a couple more minutes, so we have time for Q&A.
Okay, very quickly, one of my favorite bats, Western red bat. And they have an interesting seasonal current, so I'm going to just go through. This is the male summer range. They migrate in the winter. They occur just in the core of the Central Valley and on the coast. Finally, California migrus, which you will see throughout the Coyote Creek chain. And it forages about a foot away from coast live oaks and other oaks. So it has the same frequency as a Yuma miletus, but the best way to separate it is to just look at it foraging. It does not forage over the water. It forages, again, over vegetation about a foot away. Big brown bat are common throughout North America in houses, although they are slowly being extirpated from all our urban areas. I have caught some Townsend's bigger bats in various parts of Coyote Creek. They're cavernous roosters, so they need a cave. An old barn that's not being used, even an attic in a house, is perfect for them. And then finally, probably my favorite bat, pallid bat, which we made into a California official state bat. Took me 7 years to do that. And in the end, with the help of a 12-year-old. But this bat is, really amazing. It eats prey off the ground. It doesn't echolocate to find its food. It listens passively, and it can hear a cricket walk on the ground. I started the bat business by observing pallid bats that were picking up slugs, and I figured out that they located them by the slug eating vegetation with their radula, which makes a little sound. They occur right through the winter. We won't spend much time on this. I'm going to show you this video. So lately I've been working with a group in Israel and Germany on GPS data logging, and we're learning a lot more about the species. So we put a GPS data logger on its back. It also has a microphone on it. It's going to take off here. We're almost done, Merav. And so this is a recording that we got from one bat, presumably communicating to another bat and probably its young. And then this is the kind of spatial data that we're collecting. This is just one bat. Each one of those dots is about 1 minute apart. So it spent a lot of time in this bottom left corner. And this is some work that I did, now maybe 8 years ago on the Mexican fishing bat. Each one of those tiny circles is 1 minute apart. And some of these bats would go as much as 100 miles in a night. So it's foraging over the water. Ecologically, it's a storm petrel.
Great. Thank you very much. That was really exciting, both Jack and Dave. It was really exciting to see and hear all of the wonderful things that you've got to share with us. So thank you very much. At this time, I do have a question that came in from Ina, and she asks, she really appreciated, Jack, that you put in the lights that were interesting, and wanted to see, does the light also affect the nightlife when shining lights on them?
Well, I think the one answer is if you make it sufficiently brief, it won't. If you make it sufficiently strong and sustained, it probably will. And to know where in between, the effect cuts off. It's kind of like poisons, right? It's all in the dose. I think when we go out into the night, we try not to blast them, one organism, for very long. Many of them can see in the ultraviolet, so they can perceive the light. So we try not to overdo it.
Cool.
Can I add
Well,
something to that?
Sure.
So Ina is one of our awesome volunteers at the Newt Patrol, and what we are trying to do, and also on some of the night hikes, so we try to use red lights. Sometimes if we look at the same individual for too long, for us, for the Newt Patrol, but I think on night hikes, especially the ones that you do, Jack, you use red lights often, and I think those are less disruptive for us and for the animals. Yeah.
Oh yeah, I was just thinking about UV, but yeah, using red lights instead of white is better for the critters because many of them can't see red, and it also keeps your own night vision going because your rods do not get desensitized by red light.
Yeah, and you could do that by either adding a red tape, like a cellophane tape, on your flashlight, or some flashlights have red lights on them, so you could switch from white to red, and that works very well.
Right, that the tape, you can get red sticky tape, transparent tape from auto supply places. It's usually used to cover up broken brake lights, and it works pretty well on a flashlight. Cheap too.
A man after my own heart. I love thrift. All right, Jan has a question. Do our most common bats, like free-tailed and Myotis, stay all winter? Do they come out to feed in winter? Are some of our bats migratory?
Really good question, Jan. So Myotis do not migrate. They come out all year long, at least in our Bay Area here. Probably not every night, but they do come out. And we've been mist netting along the lower Colorado River for years. And occasionally I've done this in the winter, and we still caught, a few bats, a few Myotis. In the late fall, we often catch a hoary bat, maybe only once. I mean, only one time when we're out for the whole evening. But free-tailed bats are far more complex. And I don't know if you know much about the white-crowned sparrow migrations, but I'm guessing that Mexican free-tailed bats have something like that, because I know of very large winter roosts, bats that come in around mid-November and they leave by March, and these are roosts right by the bay, so it's cool. Not much bugs around, but that's what they want. They don't want to spend their energy in the winter if there's nothing to eat. And then there are other bats that are here all year long, and another group that have, other migrations. But in general, Mexican free-tailed bats move south in the winter, or they move to the west in the winter, and then they'll move northwards come spring, and then eastwards also to spring. So, different populations are doing different things. And I think that there's probably these, it's all one subspecies, but I have an idea these populations someday someone is going to be able to separate them and see these different clades kind of move around, but it's really complex.
Thanks so much. We have a couple more comments and questions. Amber shares that she loved learning about the bats and things that glow, and she was especially interested in bats and grateful that there was a slide that was shared showing local species of bats so she can learn more about them. Cami shares that she likes going hiking at night and doesn't use a flashlight. And then Inna asks, did the fungal white nose disease affect all species of bats?
Oh, that's a good question. No. The little brown bat and the northern long-eared bat, Myotis septentrionalis and Myotis lucificus, are especially susceptible to white nose syndrome. Myotis, a little bit. It's been recorded now in California, and even when this started 15 years ago, and by the way, it's thought to have come from Europe, and it was first introduced in Howe's Cave in New York. And I knew it'd be only a matter of time, but then on the other hand, it's a cold-loving fungus. So it likes really damp, cold places. And if it warms up, if it's dry, it can't grow, it'll die. And so most of our bats here in the Bay Area are not susceptible to white nose syndrome because they're not cave dwellers, and it's not cold and damp here.
Thanks for sharing that. Amber has a question on any tips on how folks who work in and around trees, like arborists, can spot bats in the trees. Are there any species of tree around the bay that bats are especially likely to hide in during the day?
Oh boy, absolutely. And that's the million-dollar question, because even a lot of bat biologists don't know how to approach this problem, especially if they're tree trimmers in the summer. Fan palms are not only the favored, but the yellow bat is an obligatory fan palm rooster. it roosts in fan palms. And it's been working. Naturally, they only occurred on Washingtonia fan palms in the desert. But because we plant all these Guadalupe palms and other species of fan palms, the yellow bats are working their way up northwards, just as the one of the orioles has, hooded oriole, has done the same. And Western red bat, which is a sensitive species, roosts in the foliage of mostly cottonwoods and valley oaks along riparian areas. And so, I mean, the picture that I showed you of the Western red bat was actually taken in a valley oak in Sacramento on a street tree, no less. But live oaks and oaks in general provide a lot of cavities, particularly where there's a branch that breaks and it dies back. And so you have this cavity going in, and that's often occupied by bats if it's not occupied by birds. And there's some cases where, woodpeckers have taken out bats that were in, if there's only a single bat in a cavity. Acorn woodpecker was known to take out a pallid bat. And I've seen Western scrub jays chase both Western red bats and also hoary bats. So they got disturbed during the day. In Alum Rock Park, I was leading an Audubon field trip, and we saw this. I mean, I saw this hoary bat in the middle. It was in the morning, it was daylight, and a hoary bat just went zoom, and going, oh my God, that's a hoary bat, And everybody was looking at it, and right behind it was a Western scrub jay, and it's flapping, flap, 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 flap. Well, the scrub jay, needed to see where it landed. But because that hoary bat flew so fast, it rounded a corner. As soon as it rounded the corner, it roosted in a toyon tree that's only like a couple meters high at the most. And when the Western scrub jay turned the corner, he missed seeing that, and he flew right over it.
That's a great story.
Yeah.
Jack shares that if you really want to get into bats, you might check out the annual 4-day, really 4-night bat short course at the SFSU Sierra Nevada Field Center near Yuba Pass. And in the chat, he did put a link to it. Jack, what month is that course?
I don't know. It varies. I think this June, July, and August are the main months. I mean, they have every week they change, in fact, more than once a week. But you can check it out on the website that I put there. There are lots of other good courses there.
I just got finished teaching a course on bats of Costa Rica, and it was 10 days. We only had 10 people. And then I am teaching a 3-day course on California bats at the Angelo UC Preserve, and that's September 24th through 27th. And it's through the Wildlife Society Western section. So I can put that in the chat.
Yes, please do.
I know at the Sierra Nevada Field Campus, the students go out all night and sleep all day, and they do mist netting. So
yeah, we do a lot of we do a lot of mist netting, but that's a combination course. I co-teach that with Humboldt State professor Joe Suzak, and he does the acoustic stuff, and I do the management and ecology stuff.
Yeah.
So as was mentioned earlier, Jack is a Mid-Peninsula Regional Open Space District docent who leads night hikes. So Merav says that you could see more of these lovely bats, scorpions, millipedes, and more that love the night with one of those hikes that they have. And
I can actually tell you a secret that you could request a hike from Mid-Pen, and it could be a night hike. So you could actually request a night hike on some awesome mid-pen land, and you and your group will be the only ones there because other people are not supposed to be there. And it's amazing. So
Very
just a tip.
cool.
I'd love to just make a comment. First, I want to thank both hosts. Thank you very much. And Jack, I loved your phosphorescent and luminescent talk. I have observed some species of free-tailed bats that definitely have a UV sheen to them. And when you think about bats, their world is skewed towards higher frequency. And so I believe that, they're going to be seeing much higher frequency than we are. So they're going to see into the UV light. And it could be that that's a visual signal for some of these species. I mean, I don't think anybody's reported or I don't know of any papers on that.
Yeah, I know some mammals can see into the UV. A lot can't. I don't know about bats, though.
Well, I do know that bats can see much higher than we can, and they do see UV.
Okay.
I don't know what the frequency limit is, but they are experimenting with UV lights from wind turbine towers to try and get them to avoid the turbines.
May I just add a very quick comment too?
Yes, please.
Which I didn't have time for, which is why do you get fluorescence in critters? Scorpions, all of them have been fluorescent for evolutionary very long time. Now, is there an adaptive advantage to being fluorescent? Right? And that's been an argument that's been gone back and forth for scorpions. And I think the current belief is that it is of some use, yes, because they can, the moonlight spectrum is just like sunlight, so there's UV in it, and it helps them detect moonlight better with their eyes that can detect the blue-green. Now, it converts UV that they can't see so well into. So, and they do respond to UV and get out of it because they want to hide. Otherwise, it's a hard thing to know whether some trait has an adaptive advantage. Just because your blood's red doesn't mean that was the adaptive advantage. It's really there because that's what the hemoglobin needs. It's a side effect. So who knows on a lot of this stuff.
I want to add a comment that Jackie mentioned that some bats fluoresce under UV, and I don't know if you remember, but when we were teaching our lovely Death Valley class for San Jose State, we used to do night hikes every night looking for scorpions. And a few times I saw that the little gecko, the native gecko that lives there in Death Valley, fluoresces beautifully under UV. And yeah, I was shocked to see it for the first time, and I thought, it just looked like
Cool.
Halloween decoration. But then it started moving, Anyway, it's amazing. It was really cool to see that. So I kind of wonder what other animals fluoresce, and we just don't know.
It's interesting to me that so many fungi bioluminesce, and even like the organism that doesn't reach any light. I can recall finding a fungus that had grown throughout this old log, and this was in Olympic National Park. I sat on the log not knowing it was rotten, and it crushed, and the whole log was bioluminescent. I mean, you could just pick up this rotten, and so that was never in the light. So it's as though it's some physiological purpose, retaining heat or not.
If they're in the light, where their luminescence can be seen from afar, you could imagine that it's to attract pollinators, I guess, spore spore spreaders or something like that.
Hmm.
But that's just a possibility. We do have the luminescent, bioluminescent Amphilotus, the jack-o'-lantern, western jack-o'-lantern mushroom here.
Hmm.
But I have never been able to see its bioluminescence. I once took one, well, on the East Coast, I took one, took it into a bathroom for half an hour, at night, a windowless bathroom, and sat there adjusting my eyes trying to see it. And I could barely see something. I've never seen any fungus that are bright enough around here that they would kind of attract my attention. So.
We've had jack-o'-lantern fungi here in our yard. I haven't seen them lately, but occasionally they'll be coming out of a dead tree. I'll let you know.
Yeah. I don't know. I don't know whether they have to be kind of young to be nice and bioluminescent or what. Yeah.
I think one of the real marvels of the ocean are, the bioluminescent plankton. And for those of you who haven't seen that on a boat ride or swimming or whatever, it's something that you've really got to do because it is just spectacular.
I think during the right time of year, you can go on a nighttime little boat trip in Bodega Bay and see it. You kind of go through it and you see it in your wake if you get it at the right time.
Yeah, I
Yeah,
think there was some in Santa Cruz a few years ago, right? Because I saw some posts about that. And I've seen it in Costa Rica. It was amazing.
it can be really, really thick. It usually is in Southern California. I mean, if you're on a boat, it looks like boiling lava. It's just like remarkable.
Yeah, we used to see it in Alaska when I was a kid out on the Inland Passage area.
Hmm.
The
Yeah.
boat. Yeah, it was cool, really nice.
All right. Well, let me close this out to thank everybody, and then we can resume questions if people have them. So I want to thank everyone for joining us for the Naturalist Training, where you've learned about some wonderful evening critters, bats, and UV nighttime items in nature. Thanks to our presenters, Merav Vonshak, Jack Owicki, and Dave Johnston for sharing their wonderful knowledge. They're happy to answer additional questions you may have after this close. The recording of this presentation will be available within a couple of days, and you'll receive a link in the follow-up email. In the meantime, find additional videos of our virtual BioBlitz events and training on KCCB's YouTube channel, along with our webinars.